you all for coming. Today, well, we have 3 more talks in this series, finishing up the summer. Today, Nikki Watson is going to talk. Nikki's background is in electron microscopy and biology. She worked at the Whitehead Institute at MIT for untold number of years. 20 years. Untold number of years. Before she came and joined CMS, and so we're very lucky to have her deep expertise on all things biological or electron microscopy. So today she's going to walk through some of the sort of bigger ideas she wants you to have before you come talk to her about your biological electron microscopy project. But she's located here in the SEC and has electron microscopes hidden back that way. Follow the signs to LL2301. With that, I'll dim the lights and let Nikki start.
Do we have a pointer? What's this? All right, thank you for all coming today. And as Arthur said, we've been talking about materials applications in electron microscopy. And so today, switch corners a little bit and talk about the other major application for electron microscopy, which is biological sample examination. So imaging in biology has always been probably one of the most important aspects of biology. From way back in 1964 with the first Leeuwenhoek microscope up until about the 1800s, 1960s, until we were still using good complicated light microscopes. Imaging used to be all done by hand, as the man in the top picture is showing, looking through the microscope and carefully drawing exactly what he's seeing, documenting these different biological processes and specimens that were being recorded. Then, in about 1930s, the electron microscope was developed by Ernst Ruska, and he won a Nobel Prize for his work in 1986 and then at Siemens was instrumental in getting the electron microscope as a commercially available instrument in research labs. In 1945, so not long after the microscope was actually even developed, the first picture of a cell was imaged with an electron microscope. We had a chicken fibroblasts. This is an iconic paper, the study of tissue culture of cells by electron microscopy by Keith Porter and Ernst Fulham. They're part of the group at Rockefeller Center that was working primarily to develop methods of studying cell biology with the electron microscope. You'll notice maybe in this picture that it's made up of a bunch of little pictures. So this is all done by hand on film with a film camera that has to be processed in a dark room before building all this together. There's no digital work going on at all. No computers helping you create this image. So then in 1974, George Palade was awarded a Nobel Prize for sample preparation methodologies to study cell biology with an electron microscope. And he, again, was a major part of the Rockefeller group. And his most notable discovery being ribosomes and the endoplasmic reticulum, cell fractionation. So those of you that are doing ribosome imaging nowadays with the cryo and the high resolution microscopes, you can trace your ancestry back to the very first electron microscopists of George Palade's work. And often he's described as the most influential cell biologist ever, and basically began the entire science of cell biology. So then in the 90s, we've come up with cryo-TEM. This is a new advancement in electron microscopy, especially for biology. In 2017, Jack Dubourchet and Frank and Henderson were awarded a Nobel Prize, again, for developing cryo-EM methodologies for getting high-resolution images of biomolecules. Back in 2014, Bridget Carragher and her group were the first to break the 3 angstrom barrier. So this cryo-TEM that you're doing now is really not that old. So you're part of the pioneer group at this point if you're doing this. So, but what we can learn from all these Nobel Prizes being given out for sample preparation methodologies is that there must be something special and important about biological EM that requires special preparations of your specimens. And there is, because in biology, we're studying living things. We're not studying, we want to look at things that look like this, not like this. But when we put them into a microscope, an electron microscope in particular, that's not a very life-giving atmosphere. It's in a vacuum, we're being bombarded with electrons. There's thickness issues that we have to be concerned with. So how do we get something into that environment that still looks like it was alive, which is our goal. The goal of sample preparation is to preserve the ultrastructure of the material that we're looking at in a state that looks as it did when it was alive, while at a high vacuum and being bombarded with electrons. So how do we do this? We have to think clearly before we even get started. When the cells are still happy in the incubator and the mouse is still running around in his cage, what do we want to see? How do we want to see it? And think about that before we do any fixing or preparing the tissue. So we have to define our goal. If we're looking at tissues, we want to know where that tissue we want to look at, because we're going to have to trim that very small. And this specimen preparation methodology that you choose is going to be dependent upon the specimen and the answers to the questions that you're asking. So you want to define your goals very clearly before you start any EM project. So what sort of projects, what sort of questions can we ask? First, we can ask structural questions. You've got nanoparticles, you've got purified proteins, you've got purified organelles. We can look at them and ask structural questions. We can ask morphological questions. You can have tissue cells or something of this sort, perturb it in some manner, adjust this genealogy, this genetics, see what that changes, incubate it with different toxins, see how that changes, compare and contrast. We can also look for functional studies like protein localization and by immunolabeling certain proteins with antibodies in our mildly fixed tissue. So now we can start talking about how we do this. Let's start with negative staining. It's the simplest procedure that you can do, and it's going to get you a lot of bang for your buck. Negative staining is a fast, easy, qualitative method of examining structures in isolated molecules, small cells, or purified organelles. The negative staining part is that the background is stained while your object is still transparent to the electron beam. So it appears in negative contrast. We can see things very clearly with high resolution. We've got nanoparticles of different sorts, probably lipid nanoparticles on one side. You can see that there are some divots and some structure in this image. Is that real or is that artifact? Is that because your structure collapsed? We don't really know by just looking at it. We have to do different experiments to be able to determine that. On the other side, we have, they look like full, complete round balls, so there's no collapse there. Maybe that is because the particle is solid, or maybe just more rigid. We can look at purified proteins. Amyloid fibers lend themselves beautifully to negative staining. We can even look at some whole cells, like this whole bacteria cell. In this case, we were looking for these these vesicles, extracellular vesicles that the cells are putting out. There will be possibly some damage when you're looking at negatively stained tissue because you are air drying that tissue without any air drying protection. So there may be these wrinkles on the surface of this bacteria cell, may be real. It's quite possible that the cell is ruffled. but it might also just be dried out and collapsing in the structure. It's also should be your first step when you're talking about cryo single particle sample preparation. If you take the steps to look at your samples by negative staining, looking for purity, cleanliness of the background, compatibility with buffers, these sorts of things, you're going to move forward a lot faster with your cryo-EM than if you just try to go straight to cryo-EM. The method is really pretty simple. We start with a grid, usually copper. The mesh size of the grid should be between 200 and 400. There's generally a carbon substrate or a 4-bar carbon substrate on top of the grid that will support your sample. And we like to officially ionize the grids with glow discharge to make the grids hydrophobic and allows more evenly deposited material, more smoothly diffused stain on the grid. So you don't have clumping of your samples. Then the negative staining procedure couldn't really be much simpler. The hard part is preparing your sample. That's all you in your lab going through the painstaking step that needs to get a pure sample. After that, we're just going to absorb the sample onto the grid for 30 seconds to a minute, and then pick up our 2% uranyl acetate, wash it off with 5 drops, draw off the excess stain with a piece of number 50 Wattman filter paper. We use number 50 Wattman filter paper because it's hard filter paper and we're not going to get any dust or lint from the paper onto the grids. And then we just let that air dry. And then we just, after 30 minutes, you can take it into the scope and take a look at it. So your most common stains are going to be 1 or 2% aqueous uranyl acetate, pH range of 4 to 4.5, phosphotungsten acid, 1 to 2%, and sodium and potassium hydroxide. You purify it with 1 normal potassium hydroxide. That's going to have a pH of 5 to 8, or aqueous ammonium mobilate, which is a higher pH, 6 to 7. The pH of your protein and what you're staining, and the pH of the stain needs to have some compatibility for getting good negative staining. So if your standard of 2% aqueous UA is not giving you good results, then you can try changing to another stain. It's not going to really give you higher resolution. So when people ask me what's the best one is the one that works for you. And some work better with some proteins than others. And if you're having trouble, just try a few different stains to get what works best for you. The challenges that we're going to run into, excuse me, is that you need to have a monodispersal of particles. If you've got overlapping particles, overlapping structures, you're not going to see individual particles, you're not going to see good resolution of these particles. You do need to be in a compatible buffer. This sometimes can be incompatible with the structure of the protein you're trying to image. But HEPIs, heights, UES, ideally water, all are good, good buffers to have your samples in, because they won't compete with the salts in the stains. Structural artifacts are common, as I showed you. Spherical structures can flatten or deflate. So you have to be careful about how you analyze what it is that you're seeing. Take it with some good analysis of looking at thousands of particles. The samples are air dried, and if that's a problem, if your protein's going to have a morphologically conformational change because it's air dried, or if there's other sorts of complications, you're stuck. It is air dried. The sample concentration, cleanliness, and purity are critical to getting good images. If your sample is not clean, if there's a lot of other debris, It's going to be there. You're not going to see your sample in preference to the others. It's going to be there and it's going to interfere with your ability to interpret what you're seeing. And finally, the stain by itself does interfere with the resolution of the structure. It's coating the structure. It's embedded in stain. So the fine detail is going to get lost a little bit in that the resolution. But it gets you closer to your goal of higher resolution. The benefits is that it's very high contrast, it's fast and easy sample prep, and it's easy to master. You can get going after trying it once or twice. So you can get a lot of work done quickly. All right, so other questions we can answer or investigate with bioelectron microscopy is the ultrastructure, morphological, or morphology of tissues, cells, bioengineered complexes. But we've got some problems here too that we're going to have to tackle. First off, we're going into a vacuum. Second off, tissues, cells, these sorts of things are large. And what we're looking at in a microscope has to be small, has to be thin for the beam to get through it. So the size, the physical size of the sample has to be dealt with. And the contrast of the sample. Biological material basically has no contrast. We have no color. We are really transparent things. We've got structures inside of our cells that give us contrast or give us color, but on a thin section, we're not, they have no contrast on their own. So we're going to have to deal with that. So we have to deal with the vacuum issues. We're going to have to deal by sectioning with the thickness issues, and we're going to have to stain for the contrast issues. Routine morphology is often referred to as classical electron microscopy, and it's essentially ultra thin sections of plastic embedded material. The general procedure is fixation, dehydration, embedding, sectioning, staining, and then finally scoping. Here we have a picture of toxoplasmosis. This is a parasite that infects about 40 million people. It's generally considered very non-toxic to most people, except pregnant women and immunocompromised people can suffer very, very dire consequences from the infection of this. This is a study done by the CDC's funding, because this toxoplasmosis is one of the 5 ignored parasites that need to be investigated further. So working with some people in the Laredo lab considering toxoplasmosis. You can see what we can get with the routine morphology is very high contrast resolution of this parasite. And with ultra thin sections, we can see how this parasite is filling up the cell. Lots of questions that can be answered. This is another experiment with HeLa cells showing, if you can see in the red and the blue, is the plasmalemma of the nuclear envelope, and the red is showing bleeding out of 1/2 of that the lamella. This is due to knockouts within the genome. This is also a cancer model indicating genomic instability within the cell. And this is again something that you would never see with a light microscope under this level of detail. You would not see that single lamella breaking out. So this is a project that absolutely needed electron microscopy to be able to show the effect that their experiments were affecting. For biomaterials, another example, this group had created nanodiamonds. They are 1 to 10 nanometer, or 10 to 100 nanometers, very bright fluorescent signals, unbleachable. So they had the light microscopy of these, but they wanted to show that they were actually the nanodiamonds that were lining up along the cell. So we needed to do EM because they're so small, you wouldn't see them otherwise. So that's another absolute necessity for electron microscopy. You notice in this instance too, we have the cell up here, and then this gray area here. So what we've done in this case is that we've actually left the cells attached to the petri dish so that we can see this orientation of the cells growing to the dish as well. We know the top from the bottom. So that's something to think about when you're starting to plan your experiment. What sort of orientation, what's the question I'm asking about my cells? Can I see that? How do I need to prepare them to see that? All right, so the procedure is a simple recipe. It can be mastered by anyone. We want to preserve the structure of living tissue with no alterations from the living state. So we don't want the cells to shrink. We don't want them to swell. We don't want them to have holes in them from pore infiltration. We don't want them to have holes in them from pore fixation or tissue culture techniques as well. So it should look like it did with this when it's alive, and the way we're going to achieve that is with rapid fixation. And the rapid fixation also protects the material when we get to the dehydration stage. So Karnofsky's fixative is the gold standard. You can make it fresh, ideally, in your lab. It's 3% paraformaldehyde. Paraformaldehyde is a small molecule. We use that for rapid penetration into the cell. 2% glutaraldehyde, which has 2 aldehyde groups, which is going to provide us the permanent cross-linkage of proteins. 5% sucrose and 0.1 molar sodium catalate buffer, and that is for osmotic balance. So we're not going to be swelling or shrinking the cells. This Karnofsky's fixative actually dates back to George Pilates' Rockefeller group. Karnofsky was one of his postdocs at the time. So secondary fixation and the beginning of our contrast enhancement starts with osmium. It reacts with membranes, lipids, and other nucleic material for contrast. It is a very small molecule, rapidly penetrates the cell, and but it's limited to how deeply it will penetrate into the material. So that's where we get to this size of your material you're trimming. If you are working with culture cells, that's not such a big deal. But if you're working with liver, brain, lung, something, heart, muscle, anything like this, you're going to have to trim that down into 1 inch or 1 millimeter cubes, because the osmium and your other fixatives and your other dehydration methodologies aren't going to penetrate much deeper than a half a millimeter. So get your material down to half a millimeter cubed. And again, this is where you have to start thinking about, how do I want to see this? If you're looking at muscle tissue, you want to see muscle tissue in cross-section. So you see in the cross-section of your microtubules. You want to see it in longitudinal sections so you can see all your Z bands. That has to be thought about as you're preparing it at this stage of the game. If you're looking at brain tissue, this is a difficult tissue to work with because the brain is so large. And you can get away with a little bit larger section than 1 millimeter cube if it's thinner. If you've got a half a millimeter cube, maybe you can go 2 millimeters wide. And if you're doing brain tissue, that might be where you need to go to be able to capture the level of the brain that you're trying to get at So again, we need to be thinking about our final product all the way through the process. After we've got our osmium fixed, we're going to stay with heavy metals again with uranyl acetate. So we've got the osmium staining the membranes, and then the UA staining the osmium. So we've got black membranes now for high contrast. Then we're going to dehydrate our tissue. We're going to remove all the water from the cells and eventually infuse plastic into the cells. All right, but the water and the plastic are not miscible. So we have to get the water out. We have to do that slowly. Again, we don't want to tear up the cells. We don't want swelling. We don't want shrinkage. We want it to look like it did when it was plump and happy and full of water and living in the incubator. So we do it slowly. So we commonly use ethanol, 50% ethanol water, 75, 90, 95, and then change to 100% ethanol 3 times so that you have full and complete exchange of water for ethanol. Do this on a rotary table. Keep your ethanol cold, ideally. Make sure that your tissue is fully floating in your solutions as you're doing your dehydrations. You want good surface contact with your ethanol so that you've got thorough and complete dehydration. And then we're going to start infiltrating the plastic after the ethanol 100%. We're going to bring a carrier solvent with the propylene oxide. Usually, if we can, you can't always use propylene oxide because that may reduce some of your labeling that you've added to your cells. But if you can use that, and then 100% ethanol overnight on the rotator. Sorry, 100% plastic overnight on the rotator till you're fully infused. Then you put that in a mold with a label, put it in the oven at 60 degrees overnight, at least 24 hours, 48 hours, someplace in that. The concerns you need to be thinking about as you're doing your dehydration is that you don't want your tissue to dry out when you're doing your exchanges of fluids. Very easy, especially as you get into 100% ethanol and propylene oxide, for that to evaporate very quickly. If you've got a monolayer of cells on a culture dish, that's going to evaporate very quickly. You want to avoid that at all costs. Once it's air dried, it's done. Start over. You want to make sure that you completely dehydrate your sample because the plastic is not miscible with water. If there is water left over, there'll be holes in the plastic block that you're trying to section. You won't be able to section it. You want to make sure that you use a fresh bottle of ethanol when you're doing your final dehydrations because water can be absorbed, and just that little bit can make a difference to your plastics. Fixatives, osmium, resins are all very, very toxic. Wear gloves, work in a hood. Eyewear, if is recommended as well. All of these chemicals are designed to penetrate through biological tissue very quickly, and they'll penetrate you very quickly, and they'll go right to the nucleus of your cells very quickly, and that's not going to be a healthy situation. So, and then you want to process your material in glass or polypropylene containers because the ethanols and propylene oxide can melt. So make sure that you've got your sample to something that's not going to melt. All right, after our 24 hours or 48 hours in the oven, we've got a hard plastic block, and we have to cut that into ultra thin sections for final observation on the microscope. This is probably the hardest part to learn and to become highly skilled at. Once you learn it, you've got it. It's like riding a bike, but it takes time and patience because there's 1,000 parameters that have to be right. And while I can train you how to set up and how to get going, you're going to kind of have to encounter these parameters as they come up. But the basic idea is that we've got a polymerized block of plastic with a piece of tissue in it. We're going to trim that down to a trapezoid shape with a razor blade by hand and have the tissue at the top. We're going to use a glass knife, and we're going to take the face of that block and shave it with the glass knife until it is a mirror finish, mirror smooth. No scratches or anything, perfectly mirror smooth. Then we're going to take it off the microtome, retrim the block so that our edges are perfectly clean on all four sides, again, keeping that trapezoidal shape. We are then put on our diamond knife, which has a boat that we fill with water. Water level is critical to getting this nice long ribbon of sections. We're shooting for something around 50 nanometers thin on the section thinness. Those would be silver, as you see in this picture here, they look silver. You can get away with gold, which are about 80 nanometers thick, but you're going to have better luck with silver, better images. So once we've got our block aligned to the knife and our water filled to the proper level, we can hit automated motorized cutting, and it will cut in an automated way for us and create these ribbons that we then come in with a grid and pick up we'll have 5 or 6 samples that will lay over the diameter of your grid. Your grid again should be, you can have naked grids with no substrate, or you can have a carbon film grid. The carbon film grids are probably going to work a little bit better for you when you get to the scope. You'll have less drift of the sections, especially if you are cutting really, really thin 50 nanometer thin sections. They tend to drift a bit underneath the beam. All right, so now after that, we're going to have to stain again these sections with uranyl acetate for 20 minutes and lead citrate for another 4 minutes. So the concerns that you need to keep in mind here, diamond knives are very expensive. They run about $3,000 a piece, and they're very fragile. So before you are given a diamond knife to use, you have to prove competency with a glass knife. And that takes time and practice and patience. So the basic steps again: trim the block, face the block, section the block. We're going to, as I said,
Nikki, can I just ask, what is the motivation for the trapezoid shape?
it's a more stable shape as it comes down and hits the knife. You sometimes can't do it though. If you've got a very, very small sample that barely coats the bottom of an Eppendorf tube, and it's then you're going to cut a circle. You have to get, that's what you have to do. But ideally, you've got the trapezoid that you can cut clear and crisp. All right, so then our post on-grid staining, 2% uranyl acetate, 20-30 minutes, wash, let it dry. 3% lead citrate, we're going to wash that with boiled water. We have to be very careful with the lead citrate because it's going to want to precipitate on contact with CO2, so to make a special chamber to mount your grids, stain your grids in. You want to avoid any lint contaminants. Grease from your fingers, you don't want to touch the face of your block. Anything during the sectioning and staining process is where you're going to run into contamination of your sample. And it's always going to land on your section. It's never going to land anywhere else. And if it's all over your section, if you've got lead precipitate, it can't be used. You're back starting over. Hopefully you've got enough material to just resection. But this is where your problems are going to come in. Or you can have lots of knife marks and wrinkles and all sorts of things that send you back to the microtome. So the routine morphology sample preparation is not hard. We're not heavy lifting here. We're not digging ditches, but it's unforgiving. And I guess that's where the hard part is. If you make a mistake on step 1 out of 50 steps, you're done. And you don't know maybe that you've made that mistake until step 50. So it's time consuming and it's unforgiving. Everything has to be done to a perfected level with no distractions, or else you're starting over. So what sort of problems can we run into? If we have a slow fixation, we're not paying attention to our quick fixation, then we're going to have poor membrane preservation. Incomplete dehydration, it's going to result in holes in the final block, which is going to make it impossible to section. Incomplete resin exchanges is going to cause the sections to the plastic to not polymerize hard enough, and so when you cut it, it's going to disintegrate. Or on the other hand, it's going to shatter. So you can make your resin too soft, you can make it too hard, or too hard. Dull or damaged knives are going to completely scratch the surface, so you won't be able to see anything. And the staining can precipitate all over the place. So it's frankly a miracle it ever works. So if we look at this picture that I have here, I would consider this to be a perfect micrograph. This is the goal. We have well-stained, high contrast membranes like the plasma membrane and the nuclear membrane. We can see nuclear pores. That's telling us that it's a very, very thin section. We can see the tiny little wagging little spokes of the nuclear pores. We've got good organelle preservation. In the mitochondria here, they're not swollen, they're not shrunk, they're not overly black or overly unstained. The ER is nicely recognizable. Again, we can tell the sample thickness because you can actually probably count ribosomes along the ER here. The Golgi here, if I don't know if you can see it from where you're sitting, but up close, you can see individual stacks very clearly of single membranes of the Golgi stack. So this is your goal. And when it works beautifully, it's a beautiful thing. Okay, so what other sorts of work can we do with bioelectron microscopy? So we can do functional studies and looking at protein localization by using monoclonal or polyclonal antibodies to characterize proteins and clearly define high resolution localization. If we look at the confocal picture on the right, the green is depicting Golgi, blue is the nucleus, red is ER. And that's good. That's a good confocal micrograph. You've got good clear labeling, clean, crisp, and nice in focus. But if we're studying the Golgi and we've got a protein, we know that localizes the Golgi. Is it the whole Golgi? Is it part of it? What is it? So you take that same antibody and you label the same cells, but now you look at it on EM and we say, oh, all right, so we've got a whole Golgi stack here, but that gold particle, that protein, is only localized on the very trans side of the last stack. So we've got more information that we can use now about that protein that we didn't know before. So this method that I'm going to talk about is called the Tokiyasu method. It was developed in the Tokiyasu lab in San Diego, UCSD. And it's fairly simple. We used to call this cryo-EM. We don't call it that anymore. Now it's cryo-immuno-EM. So what we do is we start with a cultured dish of cells or a chunk of tissue. We have to be very careful with our fixation at this point. Heavy fixation with 2% glutaraldehyde is going to basically make it's going to block our antibodies from our antigen more than likely. So we have to use very mild fixation. I generally use something called PLP, which is a paraformaldehyde lysine pyridate cocktail, or 0.05% glutaraldehyde, or if you're really stuck, maybe a 8% paraformaldehyde. You'll still maintain some good ultrastructure, even with 8% paraformaldehyde. Sectioning and everything else down the line becomes more difficult, but it may be all you can do, the best you can hope for. So after we've fixed, we're going to cryoprotect. So this is a 70% sucrose solution with polyvinyl pyridine in it. That makes it for easy cutting and allows us to freeze this in liquid nitrogen since it's cryoprotected. We don't really have to go to great heroics in this freezing step. As long as you are agitating the sample, it's small, then you can freeze it on a pin because of the cryoprotection. It's going to prevent ice crystallizations, which is why we do rapid freezing. So after we've frozen it, we mount it into a frozen ultramicrotome, cut frozen sections, pick those sections up with a drop of sucrose, transfer them to the grid, and then do our immunolabeling. If you do immunofluorescence labeling, it's essentially the same thing. You incubate with your primary, wash, incubate with your secondary. Secondary in our case is 5 to 10 or 6 or 12 nanometer gold particles. Wash that, stain that with neutral uranyl acetate, wash that again, and now we're embedding it. So we basically still have a high-graded sample that we want to put into the microscope. But we know we can't put a high-graded sample into a microscope. So we have to dry it some way, but we know if we dry it by air, we're going to damage it. It's going to be full of air drying artifacts. So what the Tokiyasu method basically comes down to is this methicellulose embedding. So we float the sample on a drop of methicellulose, we pick it up, we draw off the excess, we have a monolayer of this plastic that infuses the cells or the tissue and allows us to air dry without any air drying artifacts. and then we scope.
Nikki, just where do we, I assume the immunolabeling is back at temperature?
Yes. I go overnight with the primary and I'll put that in the fridge, but I'll leave it a few hours at the temperature. And then the gold is generally an hour at room temperature.
So your golden nanoparticle surface and have any modification?
Well, they're conjugated to an antibody,
Oh, I see.
right? So you've got a rabbit antibody,
Yeah,
and you get an anti-rabbit gold particle.
I see. But do you have any characterization as a density, like antibody on the nanoparticle? The fractionation?
No, that you would need cryo-TEM.
See. Thank you.
We're not looking at, we're still at a cellular level, I mean nanometer scale. All right, so here's just another example of how much better resolution, higher definition that you can, of information that you can get with gold labeling. So the top is ER, M-cherry, transected cells. And then the 3 pictures shows 3 different localizations of that M-cherry. And very clean otherwise. So here we've got one example of the M-cherry is labeling these little gray dots. And it's only in some of the ER, just where these little gray dots are. It's actually labeling the gray dots. In this one, it's pretty much in all of the lumen of all of the ER, especially in these pinched off vesicles. And then in this one, it's only in the external lumen of the ER. So it's not close to the nucleus, it's only on the outside. And you're not going to see that with immunofluorescence or confocal or super resolution. All right, so immuno-EM, there's conditions that have to be satisfied. First, you have to determine the level of fixation. The antigenicity of the target molecules, can it be preserved or is it going to wash away? Is there enough antigen present in a 50 nanometer thin section? So you want to do your whole cell immunofluorescence, but you really do yourself a favor if you do semi-thin immunofluorescence and see if you can still see your fluorescent antibody. If you can't see it in a half micron thick section, you're probably not going to see it in an ultra thin section. Is your antibody clean, specific, concentrated? Will GFP work, and is that clean, specific, and concentrated? There are a lot of good anti-GFP antibodies available now that work beautifully on EM, anti-M-cherries. So there's a lot of progress there that wasn't available even 10 years ago. You need to think about your positive and negative controls. You think negative control is going to be just a secondary without the primary. See what your background is. Positive control is harder to come by unless you do this sort of work for a living in your lab. It's harder to have a constant everyday winner that you can say is your positive control. But your negative control is imperative. You have false positives all the time. So, and then finally, do you have good sampling? When we're planning our experiments, whatever it is, if it's immuno-EM, if it's negative staining, if it's classical routine morphology, you want, I can't emphasize enough to plan your experiment. Plan it carefully. You want to know, have an idea in your head what your final outcome should look like, because that's going to depend about how you do your embedding and everything else. You want to have a defined question. The problem, I think, most people do, at least in biology with my interactions, is they assume I can see my results, therefore I'm going to be able to interpret my results. And that is really not generally true. Biology is diverse. Cell-to-cell diversity is going to confuse your results. So you want to make sure that the questions you're asking are, is it there or not? Simple, yes, no. Did this happen or not? Don't try to get complicated thinking that you're going to be able to interpret the landscape, because it's just going to be too confusing. You want to avoid a needle in a haystack. If you've got a cell culture situation, you've got 10 cells out of 1,000,000 that are showing your phenotype, stick to light microscopy, where you will never find those 10 cells on electron microscopy. If you want to do EM on these cells, you've got to get it up to at least 70% of the cells showing your phenotype. Tissue culture, so we don't want to, so again, we don't want to look for a needle in a haystack. We're not going to find it. It's not going to happen. And you can't trust it that it's a needle when you do find it. It could just be some abnormality cut in the right direction. But if you do have a situation, there are certain sorts of research that are just by definition needles in a haystack, embryological work, developmental work, brain work. You're looking for a single cell. If you're going to have a needle in a haystack situation, then make it a very, very big needle, a needle that you can't avoid. That when I look at it with low magnification, it's enormous and I can find it. So that's how you get around that problem. So I'm just going to talk just real quickly about scanning EM. This can also be used very effectively with biology. It's a relatively quick and easy procedure that you can get done in a day and be imaging the next day. Again, fixation, same fixation you use with TEM, dehydration, and a graduated process. Critical point drying, because we want to have an air dried sample, again, with no air drying artifacts. Critical point dryer will get us there by exchanging ethanol with CO2, bring CO2 up to a gaseous state under pressure. Now we have an air dried sample that hasn't had any air drying artifacts. Mounted on a stub. Coat it with a heavy metal. What's going on? And then look at the scope. And again, here's some beautiful examples of how you can really enhance, get the cover of cell. How do I get rid of that? Nick, do you know how to get rid of that?
Can you move the window? Can you tag it? If not, try exiting the PowerPoint presentation. Hit like escape and then try to relaunch.
Drag and move it somewhere else.
I don't see it on my. So this is a study with the Arabidopsis looking at different pollination strategies that this Arabidopsis has. In this one, Loretta, the thistle is ready for pollen, but the anthers have no pollen present, so this is not going to be able to self-pollinate. And this is the opposite. The pollen is ready and the thistle is ready, so this is self-pollinate. Simple demonstration of a complicated system. We also have available cryo-SEM. And this is a method of preserving structures or physical states by rapidly freezing that cannot be preserved or may be damaged by chemical fixation. So you start with a live material, you put it in the Leica instrument, freeze it, works the magic, and then that pops right into the SEM. So it's not an easy procedure, but you don't have to go through all the chemical fixation. All right, and then we'll talk a little bit about cryo-TEM. Cryo-TEM is for whole cells, viruses, proteins, cryo-electron tomography, single particle, and electron crystallography. The procedure, again, is relatively simple in theory. It's difficult in practice because you're working under liquid nitrogen temperatures. Everything is freezing, and things can get damaged very, very quickly with the slightest bit of lack of focus on what you're doing. So, but basically, we're going to prepare our sample that you have determined its concentration and quality, hopefully by negative staining, so you know what is in your test tube. You want it to probably be 10 times as concentrated for cryo-TEM than it would be for negative staining. You're going to put a drop on a grid and then in a controlled manner in the Vitrobot, you're going to blot it dry, you're going to drop punch freeze it, transfer it into a grid box, which then can be transferred into the microscope. So it's rapid fixation of hydrated samples. You're going to be looking at the sample basically in vitrified ice, which is ice with no crystals, and hopefully it's a monolayer of those particles. Imaging and analysis. The take home is that you're taking 1,000 pictures of 1,000 objects and averaging them together in some manner computationally. So, but that's easier said than done. So first, we're going to collect all our images. We're going to align those images. We're going to particle pick and correct for any defocusing if we need to. You're going to categorize those particles. And then through software, you're going to run through many iterations of those categorizations until you get a refined, nice high resolution image. All of this takes practice and patience, again, that you don't over interpret or over iterate your images, that you don't under process them. You're going to then fit that to a model and then get your final 3D structure. The concept is not difficult. It's in practice and it's time consuming. And of course, the protein that you're looking at may make it even harder because it won't freeze well. So patience. All right. So the most difficult aspects of biological electron microscopy is going to be the sample preparation of their specimen material. Once a number of these techniques have been mastered, the beginner can easily be trained to use the electron microscope. As we've heard in this series so far, the microscope's doing the work. You're getting your data based on interactions of materials with the beam. In biological electron, we're not doing that. The microscope is simply our camera. We're just taking pictures. So there's nothing fancy that we need to know about the electron microscope except how to focus well, how it operates. But we're not doing magic with the microscope. The magic happens before we get there. So remember, define your question, choose the correct sample preparation, and determine your controls. All that's going to help you interpret the final results. So for training and services at CNS and here in SEAS, there's 3 methods that we can go about for routine morphology. Negative staining, we're going to train you to do your negative staining and train you to use microscope for the most part. Unless, it can be a service, but it's probably easy enough not to. For routine morphology studies, the service is going to be an average of 2 weeks turnaround time if I'm doing the imaging for you. Training is going to be 4 to 6 months because sectioning is difficult. And it takes dedication, it needs practice. You have to sit there and do it. For negative staining, structural studies, the service would be a 1-day turnaround time. You give me samples, I'll give you pictures probably the next day. Training is going to last 1 to 2 hours, less if you already know how to use the microscope. Immunoglobulin labeling, if we're going to do that as a service, you do have to plan 1 to 6 months, would not be out of line from beginning to end. There's a lot of parameters that need to be worked out. They all have to be worked out individually, from fixations to antibodies and all sorts of things. Training, I don't know, if you're already a good electron microscopist, training is an option. If you're not, you might want to spend your time doing something that's more productive with your time. All right, so just in conclusion, there are few biological problems that do not benefit from the use of electron microscopy. So think about it in your work, that electron microscopy can add to the overall product that you're publishing. If you're interested in including EM analysis in your research, just contact me for a consultation and we can talk about if it's practical and how to get it done. And if you do contact me, please bring any supporting information that will help me understand your questions and help us design an experiment that's going to work best. And my email is at the bottom and it can be found on the CNS website too. All right, so that's it for me. Thank you very much.